Okay, guys. Um, this is going to be another video podcast, chapter 25. And what we're going to be talking about here are the chemical reactions that happen inside cells in order for them to generate immediate usable energy from the food that you consume. Just as an overview, uh, y'all know instinctively that cells can't directly use large foodstuffs, you know, for instance, that you would ingest, like a bran muffin or a hamburger patty, directly as a source of fuel. What has to happen prior to that is that the food has to be mechanically and chemically broken down in order for it to be absorbed into the cells. And we went through that in chapter 24, the process by which we take these large chunks of food, we break them mechanically into tinier and tinier pieces, and then we chemically break them all the way down into what we call monomers, which are the simplest chemical components of macromolecules that can be absorbed into the bloodstream. So, by doing that, what we're able to do is move the chemical potential energy that's stored in the food you eat into the cardiovascular system. And what we're going to talk about today is what happens after that occurs. How is it now that we get those monomers that are in the blood into cells for them to use to generate ATP? Remember that we nicknamed ATP biological gas in the tank, and it's a source of energy that is immediately available for the cell to do work. Now, what do we define as work? Movement, transport across membranes, the building of large molecules, anything that requires an energy input in order to happen that wouldn't happen spontaneously on its own. Okay, so this is what we would call in chemistry the driving of an unfavorable reaction. Basically, we're trying to tip the energy scales in our favor. So, we'll define metabolism and we'll explain why cells have to synthesize new organic components. So, what you're looking at here is a diagram of the cell membrane, the cytoplasm, and the mitochondria. And this is sort of an overview of the different chemical reactions that happen in each of these different compartments. Cells are chemical factories. They break down organic molecules to obtain energy, and they can then use this energy directly to generate ATP. Mitochondrial reactions provide most of the energy that a cell needs. To metabolize, cells have to have a supply of oxygen and nutrients, including water, vitamins, mineral ions, and organic substrates. Now, if we look at each of these, what are important about each of these components? Well, the oxygen's important because without the oxygen, we wouldn't be able to extract as much chemical potential energy from organic fuel as we would in the absence of oxygen. Metabolism in the presence of oxygen is called aerobic cellular respiration. And you generally get between 36 and 38 ATP per glucose molecule if there's oxygen present. And we'll see exactly why that is towards the end of the lecture. Without oxygen, you can only manage 2 ATP per glucose molecule. And so you can see that you don't get nearly as much an energy payoff without oxygen as you do with oxygen. Oxygen aids in what we would call metabolic combustion of these organic fuels. Now, water provides an environment inside which chemical reactions can occur. Without an environment in which molecules can move, there wouldn't be any potential for molecules to collide, and collision is a prerequisite for a chemical reaction to occur. So this is our environment in which all these other reactions we're looking at actually take place. Vitamins and minerals, well, what are they? Well, in terms of metabolism, they are like tools that enzymes use in order to make chemical reactions go. If an enzyme, for instance, was an electrician, his toolbox would be his vitamins and his mineral components. The organic substrates are actually where the chemical potential energy is located. And long story short, the money bond in biological systems is the carbon-hydrogen bond. There's more chemical potential energy in the carbon-hydrogen bond than any other bond in the macronutrients that you consume. Oxygen is absorbed at the lungs. The other substances are obtained by absorption across the GI, primarily in the small intestine. The cardiovascular system then carries these substances through the body. We talked a little bit about how life has changed on this planet as we've been going through each of the body systems, and we noted the fact that early on in evolution, life was a lot simpler. We were single-celled organisms, and we got food essentially by transporting it directly across the cell membrane, and then we excreted the wastes in the reverse fashion. But as the earth became more complicated, as the environment changed, parts of the earth got colder, parts of the earth got drier, and that presented a challenge to life on this planet. Life had to adapt or die. And one of those adaptations was to go from a single cell to a multicellular organism. And when that happened, what you had was an arrangement of cells and tissues, some of which were distant from an oxygen source and a nutrient source. And so we had to develop a delivery system to get them the fuel and the oxygen they need in order for them to live. And so that's why the GI, the respiratory, and the cardiovascular system all have to work together in order to provide these vital components for life. These substances diffuse from the blood into the tissues where they're absorbed and used by the cells. The mitochondria break down organic nutrients to provide energy for cell growth, division, contraction, secretion, and other functions. Basically, the life processes. Metabolism refers to all the chemical reactions that take place in an organism. There's tons of chemical reactions going on in every cell in your body right now. Cellular metabolism provides the energy needed to maintain homeostasis and to perform essential functions, those being metabolic turnover, the periodic breakdown and replacement of cellular components as they wear out, growth and cell division, and processes such as secretion, contraction, and the propagation of action potentials. All the cell's organic building blocks form a nutrient pool that the cell relies on to provide energy and to create new cellular components. Essentially, these are all processes that would not happen spontaneously. So they need some source of fuel to draw on in order to drive them to completion. And that's what is contained in the chemical bonds in the macronutrients that you consume. And as those bonds are broken, that energy is seized and then transferred to other molecules and eventually to ATP, which is used directly to drive these processes. The breakdown of organic substrates is termed catabolism. This releases energy used to synthesize ATP and other high energy compounds. Mitochondrial ATP provides energy to support both anabolism and other cell functions. Now you might ask, well, what the heck is anabolism? Well, anabolism and catabolism are two broad categories of chemical reactions that make up all of metabolism. When we're talking about anabolism, we're talking about building large molecules from smaller pieces. And that's, again, not a process that would happen spontaneously. That needs an energy source in order for it to drive it to completion. Just think in terms of building a large structure, right? You build a large structure from smaller pieces, and in order for you to assemble those pieces, you have to put energy into the system in order to produce your finished product. The bricks, the mortar, the paint, all that don't just spontaneously assemble themselves. And the same is true at a molecular level. Monomers don't spontaneously assemble themselves into macromolecules, which can then be used by the cell for a whole host of other purposes. They need some other energy source to draw on, and it's by the combustion of that energy source that we liberate enough energy to drive these normally thermodynamically unfavorable processes. Okay. Now, catabolism is the breakdown of large molecules with the intent primarily of liberating the energy that's locked up in those chemical bonds so that we can use it to perform work. So anabolism relies on catabolism in order to operate. One wouldn't occur without the other. And what you're looking at here is a diagram showing you a mitochondrion, and this is showing you some of the molecules that can feed into the metabolic pathways that take place inside the mitochondria. Anabolism is an uphill process that involves the formation of new chemical bonds. Cells make new organic components for 4 reasons: to perform maintenance and repair. All cells have to use energy to perform maintenance and repair because most cellular structures are temporary. Their removal and replacement is part of metabolic turnover. Essentially, any macromolecule in a living system has a finite lifetime. And once it's served its purpose, it needs to be replaced, and that requires energy. It's also used to support growth. Cells that are preparing to divide increase in size and make extra proteins and organelles, and that manufacturing process also requires energy. And then they also use it to produce secretions. Well, what are secretions? They are molecules that are assembled from component parts, which are then sent into the extracellular environment, generally by some sort of active transport, which again is an ATP dependent process. So actually secretion relies on chemical potential energy in 2 facets. It relies on it to make the secretory product, but it also relies on it to propel the product into the extracellular environment. Neither one of these things would happen spontaneously. It also needs energy to store nutrient reserves. Most cells prepare for a period during which they're not going to have a ready source of energy in the extracellular environment. This is an emergency time, an interval of extreme activity, or a time when the supply of nutrients in the bloodstream is low. And so what they do is they, within the cell, have what are called inclusions, which are storehouses of chemical potential energy that they can draw on in between these periods of low nutrient delivery from outside environment. The most abundant storage carbohydrate is glycogen, which is a branched chain of glucose molecules. The most abundant storage of lipids are triglycerides. Now, in the human body, most of our energy stores are in the form of lipid, and we've kind of touched on why that is already in the previous weeks, but just to revisit that, if you look at the typical triglyceride, what you find are a couple of features in these molecules that make them ideal as a storehouse of chemical potential energy. Number one, they're hydrophobic, and what that means is that they tend to exclude water and as a result take up a minimal volume. They don't have to be hydrated in order to be stored. In addition to that, if you look at the number of carbon-hydrogen bonds in your typical triglyceride, which is one of the many forms that a lipid can take, it's 20 to 50 times more than the number of carbon-hydrogen bonds in your typical carbohydrate or your typical amino acid. Okay, we're talking here about monosaccharides and individual amino acids. And so what you're gaining is a huge chemical storehouse that takes up minimal volume and has maximum energy storage. And that's why we tend as a species to favor the storage of lipids when we're not actually using those calories to do work. Proteins perform a lot of vital functions for the cell, and when energy is available, cells will manufacture them. The nutrient pool is the source of the substrates for both catabolism and anabolism. There is an energy cost to staying alive, even at rest. All cells must use ATP to perform maintenance, remove and replace intracellular and extracellular structures. In addition, cells have to spend energy performing functions such as growth, secretion, and contraction. They're really, this term at rest is really a misnomer, because even when we are technically at rest, the tissues are still performing maintenance functions that require ATP. When you're asleep, for instance, you're still breathing, muscles are still contracting, ATP is still being used to do things like maintain membrane potential, send nerve signals, excrete waste, processes that are constantly ongoing, housekeeping processes, as it were. So there's always an energy cost as long as you're living. You are depleting your energy reserves. And as a result, we always need a constant influx of new chemical potential energy. So let's talk about carbs first. We'll describe the basic steps in glycolysis, the TCA cycle, and the electron transport system. And these are what are termed the big three metabolic pathways that generate the ATP needed to make life processes occur. And they rely on each other. They are interconnected. They happen in physically different locations inside the cell, and we'll detail where those are. And we'll summarize the energy yield of glycolysis and cellular respiration. So, most cells generate ATP and high energy compounds by breaking down carbs, especially glucose. The complete reaction sequence can be summarized as glucose plus oxygen gives you carbon dioxide and water, and then what isn't shown here, chemical potential energy in the form of ATP and heat. The breakdown occurs in tiny steps, several of which support the conversion of ADP to ATP. The complete breakdown of one molecule of glucose provides a typical body cell a net gain of 36 ATP. Although most ATP production occurs inside mitochondria, the first steps take place in the cytosol. Glycolysis breaks down glucose in the cytosol and makes smaller molecules that can be utilized by mitochondria. Because glycolysis does not require oxygen, the reactions are termed anaerobic reactions, and meaning without, aerobic meaning oxygen. The subsequent mitochondrial reactions have to have oxygen to take place and are termed aerobic respiration. This is called cellular respiration or cellular aerobic respiration. Now, if you look at the term glycolysis, it literally means sugar, that's the glyco portion, and lysis means to split. So what happens in glycolysis is that you split sugar in half. And in the process of doing that, you perform a little bit of chemical extraction of potential energy, and you make a net gain of 2 ATP per glucose molecule. That's the only step of respiration that can occur in the absence of oxygen, and that's why I was trying to make the point that oxygen is vital for the production of sufficient ATP to drive life processes, because without it, you can only run this initial step. You cannot run the citric acid cycle and the electron transport chain effectively. Glycolysis is the breakdown of glucose to pyruvate. Pyruvate is a 3-carbon molecule. Glucose is a 6-carbon molecule. In glycolysis, a series of enzymatic steps breaks the 6-carbon glucose into 2 3-carbon molecules of pyruvate. Inside cells, each pyruvic acid loses a hydrogen ion and exists as pyruvate, which is the anion of the acid, which has now donated its proton to the aqueous surrounding environment. And that what the net effect of that is to lower the pH. Glycolysis requires glucose, appropriate cytoplasmic enzymes, ATP and ADP, inorganic phosphate, and NAD, which is a coenzyme that removes hydrogen atoms during certain enzymatic reactions. Now, really, when they say hydrogen atoms, what they're talking about is a proton and the electrons that it carries. And it's the electrons that we're really interested in here, because it's their change in location that is going to ultimately release energy to other molecules that will eventually be used directly to do work. So let's look at this reaction sequence in detail. If any of these are missing, glycolysis can't take place. It begins when an enzyme phosphorylates the last carbon atom of glucose and creates a molecule called glucose 6-phosphate. So what's happening here at the cell membrane is that glucose is being delivered by the bloodstream. And in order to transport the glucose across the cell membrane and into the cell, there's a sodium glucose symporter which will take the glucose in along its concentration gradient, and then the first reaction in glycolysis is to phosphorylate this molecule, and by doing that you essentially trap it inside the cell so it can't go back out. So once glucose becomes glucose 6-phosphate, there's no way for it to diffuse back out into the bloodstream. So that is a way to basically start the ball rolling here. This step costs the cell 1 ATP molecule, but it has two results. It traps the glucose inside the cell because phosphorylated glucose cannot cross the cell membrane, and it prepares the glucose for the rest of the reactions in the pathway. A second phosphorylation takes place in the cytosol before the 6-carbon chain splits into 2 3-carbon fragments. Energy benefits begin to appear as these fragments are converted to pyruvate. This anaerobic reaction sequence gives the cell a gain of 2 ATP for each glucose converted to 2 pyruvates. Glycolysis yields an immediate gain of 2 ATP per glucose that it breaks down. The ability to catch that energy depends on oxygen. If oxygen supplies are adequate, mitochondria absorb and break down the pyruvate and generate even more ATP. The hydrogen of each of the pyruvates are removed by coenzymes and are the source of most of the cell's energy gain. The carbon and oxygen atoms are released as carbon dioxide during decarboxylation reactions. Now, every time you see a carbon dioxide molecule produced at any of these metabolic steps, that essentially is a massive release of energy from the molecule that was oxidized. And that generally results in the production of a high energy molecule. Could be ATP, could be NADH, could be FADH2. In biochemistry, carbon dioxide is termed a stable leaving group. Its formation is thermodynamically favored, and as a result, anytime there is an opportunity to form a carbon dioxide molecule, it'll often happen spontaneously, and that will result then in the release of energy that can be trapped by other molecules and used to synthesize these high energy intermediates. Basically, the enzymes trap the energy and then turn around and transfer it to a molecule that's going to hold it temporarily. Two membranes surround the mitochondria. The outer membrane has pores that are permeable to ions and pyruvate. Ions and molecules are thus easy to enter the inner membrane space. The inner membrane contains carrier proteins that move pyruvate into the matrix. Now, what you're looking at here, if we zoom in on this series of reactions, is the initial steps of glycolysis, okay, the production of pyruvate, and then the transfer of pyruvate into the mitochondria, okay, and at that point, what happens is that we generate a molecule of something called acetyl-CoA, and it's the acetyl-CoA that actually directly enters the Krebs cycle, which we're going to talk about on the next page. But what we've accomplished up to this point is we've split the sugar in half, okay, and we have extracted some chemical potential energy from it. The way that we're able to determine that is to look at the end products of glycolysis, which are two molecules of pyruvate, and count the number of carbon-hydrogen bonds in a typical pyruvate molecule compared to a typical glucose molecule. And what I'll do here is we'll do a little art. Okay, so let me grab the pen here, and let's make it a black pen. And let me draw up here a molecule of pyruvate. Okay, pyruvate essentially looks like this. I'm using chemical shorthand here for pyruvate, and I'll explain to you what I mean by that in a minute. Okay, so that's pyruvate. And then down here, I'll draw a molecule of glucose. Okay, so that's molecule of glucose. Now, this is organic chemists use this as a way to that's an OH there. It's a little hard to see, but they use this as a way to quickly draw complex molecules without having to draw every carbon-hydrogen bond. But essentially, if we count the number of carbon-hydrogen bonds in a glucose molecule, there'd be 1 here, 2 here, 3 here, 4 there, 5 here, 6 and 7 there, okay? And that is 7 carbon-hydrogen bonds in a typical glucose molecule. Now, if we look at a pyruvate molecule up top, okay, we count a total of 3 carbon-hydrogen bonds here. Now, we make 2 molecules of pyruvate when we send it through the Krebs cycle. And remember what I told you about determining the calorie content of organic molecules. Basically, if you count the number of carbon-hydrogen bonds, that'll tell you how many calories are in that molecule. And what you can see is that when you look at the 2 molecules of pyruvate up here that you would generate at the end of glycolysis, you have 6 carbon-hydrogen bonds, whereas you have 7 down here. And so what that means is that we've lost some chemical potential energy as a result of going through this pathway. And we've used that to generate now 2 molecules of ATP. So what's happened here is an oxidation. Now, oxidation and reduction are kind of tricky concepts. Essentially, in biological systems, when we're talking about an oxidized molecule, we're talking about a molecule that has less chemical potential energy than a reduced molecule. And so now you might ask me, well, how do I tell when a molecule is oxidized and when a molecule is reduced? Well, there's a real easy way to do it. The molecule with the most carbon-hydrogen bonds is going to be the molecule that is the most reduced. And the molecule with the fewest carbon-hydrogen bonds is going to be the molecule that's most oxidized. And so that's a direct way of doing sort of an accounting of the total energy content in that molecule. So when you see these molecules in your book, and there's a video that I've got posted on Blackboard that runs through these pathways. If you look in the chapter 25 lecture section, There's a real good video on cellular metabolism that I recommend that you watch. But it will show you what these molecules look like. And essentially, all you have to do is, if you count the number of carbon-hydrogen bonds, that'll give you an indication of the energy content of the molecule. So what we can see as we've moved through glycolysis is that we've lost some energy from the substrate, okay, from glucose. And where's that energy gone? Well, it's gone to make 2 ATP molecules, and it's also gone to make a molecule of NADH, okay? And that molecule is generated at this step, okay? Now, as far as anaerobic respiration is concerned, the immediate energy payoff are the 2 ATPs that we generate directly. The cell can immediately use that to do work. The NADH represents still potential energy. We haven't converted that into ATP until we actually oxidize this molecule in the subsequent steps of aerobic metabolism, which we'll get to here in just a minute. So this is why I've been stressing the point that the immediate energy payoff is just these two steps up top here, this ATP generating step and this ATP generating step. Remember that you send 2 molecules down this branch of the pathway. Okay, so you lose 2, but you make 4 for a net gain of 2. Okay, and we can see in the end product that we've lost some chemical potential energy. This is pyruvate up here, glucose down at the bottom. Okay, now the next step is to send pyruvate into the mitochondria. And what happens when that occurs is that a CO2 molecule is lost. Now remember what I told you about CO2 as a leaving group. It's an extremely stable leaving group. So when you see CO2 come off, you can always assume that energy's been liberated and is going to be used at this biochemical step to do something. And several things happen at this step, right? One thing that happens is that we form a molecule of NADH. Now, NADH is a high energy compound that's eventually going to be oxidized and used to generate ATP. So we use some of that energy to form NADH. We use the rest of the energy to form a molecule of acetyl-CoA by forming a covalent bond between the acetyl group of the pyruvate and coenzyme A, which is contained inside the molecule that actually forms now this starting product for the Krebs cycle. And this is all happening inside the mitochondria in the matrix, which is basically like the cytoplasm of the mitochondria, the innermost core. So in the mitochondria, pyruvate participates in a reaction involving NAD and CoA, which yields a single carbon dioxide, 1 NADH, and 1 acetyl-CoA. Now remember that you make 2 pyruvates for every glucose that you split. So you're going to run this cycle twice. We're just taking 1 pyruvate through this trip right here. This sets the stage for a sequence of reactions called the TCA cycle. In the first step, an acetyl group is transferred from CoA to oxaloacetic acid, and this produces citrate. And this is where the cycle gets its name from. Citrate is the anion of citric acid. TCA stands for the tricarboxylic acid cycle or the citric acid cycle. The Krebs cycle is also used as a description of this biochemical pathway. So you got 3 names for the same set of biochemical reactions. The function of the citric acid cycle is to remove hydrogen atoms from organic molecules and to transfer them to coenzymes. And again, when we're saying hydrogen atoms, we really mean electrons that are associated with the carbon-hydrogen bond. So what's going to happen as we walk through this pathway is we're going to very slowly oxidize this pyruvic acid. And the way that we know that we've oxidized it is that we're producing now the most oxidized form of carbon that you can get, which is a carbon-containing compound with no carbon-hydrogen bonds in it, and that's carbon dioxide. These CO2s that I'm pointing to here represent an energetically exhausted metabolic endpoint, and there's no biochemical energy that can be extracted from these molecules. So this means that the energy that was in this pyruvate molecule has now been transferred somewhere else. And the place that it's been transferred to is to these intermediate molecules, NADH and FADH2, and also directly to make ATP in one of the steps. CoA is released intact and then can bind another acetyl group. A complete revolution of the citric acid cycle removes 2 carbons and regenerates a 4-carbon chain. The entire sequence can be summarized as acetyl-CoA plus 3 NAD plus FAD plus GDP plus inorganic phosphate plus water gives you CoA plus 2 carbon dioxides plus 3 NADH plus FADH2 plus 2 protons and a GTP. Now, I want you to notice something about this equation, okay? Where is the high energy molecule on the left side of the arrow? It's the acetyl-CoA molecule, okay? Where are the high energy molecules on the right side of the arrow? The NADH, the FADH2, and the GTP, okay? These represent temporary energy stores that are eventually going to be transferred directly to ATP, okay? So we've moved energy from one molecule to another molecule. Now, in the process of performing all these steps, some heat is generated. That's a byproduct of any chemical reaction in a living system, is the generation of some amount of heat. That heat is forever lost as usable energy, but remember that physiologically it has a purpose. Enzymes operate at optimal temperatures, and the optimal temperature for the enzymes that make up metabolism is 37 degrees C, which is 98.6 Fahrenheit. That's body temperature, okay? And so the heat that's generated here serves to maintain the optimal temperature for these enzymatic reactions to proceed. The only immediate energy benefit of one revolution of the Krebs cycle is this single GTP that's generated right here. It's immediately used to make an ATP molecule, which can be used instantaneously for work, okay? So again, without oxygen, all right, we've just got those 2 ATP from glycolysis and these 2 ATP that are generated down here. All the rest of the chemical potential energy at this point is in NADH and FADH2 molecules, and we don't get the energy payoff from those until we get to the electron transport chain. Now, the intermediates are actually shown in this graphic, and what I want you to notice about them is that what essentially is occurring as we go through each of these intermediates, these Krebs cycle intermediates, these are all carbohydrates, okay? But every time we strip off a carbon dioxide molecule, you can see that we produce some sort of high energy intermediate that it's going to be used later. For instance, here, when we liberate a carbon dioxide off of isocitrate, we form an NADH, okay? That is a high energy intermediate that will eventually be oxidized to generate ATP. Down here, when we go from alpha ketoglutarate to succinyl-CoA, what happens here is that we lose a carbon dioxide molecule, and that energy's liberated to form another NADH, okay? So 2 high energy payoffs here, okay? Another decarboxylation is up here, okay, at the entry point into the cycle where pyruvate is introduced to the citric acid cycle as it joins to oxaloacetate. That decarboxylation is used to produce an NADH molecule, okay? So every time you see a CO2 come off, you think energy payoff, okay? The rest of the reactions where we generate high energy molecules usually involve some other form of oxidation. The transfer of succinyl-CoA to succinate is an energy-yielding reaction. That is an oxidation, okay? And the transfer of succinate to fumarate is an oxidation reaction, which generates FADH2. And if you look real close at this graphic, you can turn to the chapter in your textbook to get a bigger picture of these intermediate molecules. You can just, as an exercise, okay, count the number of carbon-hydrogen bonds, say, between succinate and fumarate, and what you'll find is that there are fewer in fumarate than there are in succinate. And there you go again. The number of carbon-hydrogen bonds is a direct correlation to the energy content of the molecule. Okay, so that's the way that you can tell what molecule has more energy, and as a result, what molecule is more oxidized and what molecule is more reduced. The more reduced the molecule, the more chemical potential energy that it has. Okay, the last step of the big three is glycolysis, also known as oxidative phosphorylation. It's the generation of ATP in mitochondria in a sequence that requires coenzymes and consumes oxygen. And this makes about 90% of cellular ATP. So this is really where we get the bulk of the energy payoff in all of metabolism. And it looks a little daunting when you look at the graphic here, but essentially this works in a very similar way to a hydroelectric plant. I don't know if any of you have ever been to Las Vegas, but the first thing you think of when you think of Las Vegas, besides losing money, is the generation of lots and lots of electricity to run all those lights out there. And have you ever wondered where the electricity to run Las Vegas comes from? It comes from a hydroelectric dam. Essentially what they've done in Las Vegas is that they've dammed up a river, and they have used the water that builds up behind the dam to turn turbines inside the facility. And these turbines essentially are wheels that have magnets on them. Okay, and when these magnets rotate at a high speed, they're situated around large copper coils. So you think of the coil as like the spoke of the wheel, and think of the turbine as the wheel itself, and the magnets are like the tread, if that makes it easier for you to visualize. So as the water flows through these turbines and this wheel spins, these magnets rotate at a high speed around these copper coils, and it's a peculiar property of magnetic fields that when they rotate around a piece of metal, they generate an electric current. That's where the electricity comes from. And so what you're seeing there in a hydroelectric plant is the transformation of one type of energy to another. Basically, the potential energy, which is the energy of the water building up behind the hydroelectric dam, is transferred into kinetic energy as the water falls from a high point to a low point. That kinetic energy becomes kinetic energy that turns the turbine. Okay, as the turbine is spinning, that kinetic energy is transformed into electrical energy as the rotating magnetic field generates the electric current inside the copper coil. And then that electric energy can be used to be any other kind of energy, right? It could be light energy, heat energy, kinetic energy, if you want to run an electric motor, for instance. Almost anything that you could think of that you could drive with an electric current. Okay, so energy can change forms, okay, and that's exactly what's happening here in the electron transport chain. Is we're taking different forms of stored energy and we're transferring them from one stored form to another stored form. Okay, and with every change, with every energy transformation, you lose a little bit of that energy as heat. That's inevitable. Okay, and that's what serves to further randomize the universe that we live in, right? That's what produces an increase in entropy, the second law of thermodynamics, right? Which I like to restate as things take the easy way out whenever they can. Okay, so let's look at this sequence of events here. This is a drawing, a schematic showing you the enzymes that are involved in the electron transport chain. Now, what you'll notice here is that the Krebs cycle reactions are happening here in the matrix, okay? And what's being generated in the Krebs cycle, besides ATP directly, is large amounts of NADH and FADH2, which represent now high energy molecules. They're in a reduced state. And what we're going to do is we're going to oxidize them. Now, when we oxidize them, what we're essentially doing is we're stealing electrons from them, okay? And when we steal electrons from them, they get oxidized, and the thing that steals the electrons gets reduced. And so here you can see NADH. When it interacts here with the first participant in the electron transport chain called FMN, it becomes oxidized to NAD. Now you might say, okay, well, what happens to the NAD now? Well, it can be used in subsequent Krebs cycle reactions. But where did those electrons go, those high energy electrons in this molecule? Well, they get swiped by FMN, and then they are passed from this electron carrier to the next electron carrier in the chain, which is coenzyme Q. Now, what happens when this occurs is that the electrons change their location. It's sort of like, you could think of them kind of like as falling down a staircase, okay? As you go from the top step down to the bottom step, you're losing a little bit of energy. You're losing chemical potential energy in this case. And so as these electrons lose chemical potential energy as a result of their change in physical position, that energy is captured and is used to pump protons that are generated by the oxidation of these high energy molecules, NADH and FADH2, which are pumped into the inner membrane space, which is this area between the outer and the inner mitochondrial membrane. And so what happens is that this zone becomes acidified. Okay, a lot of protons get pumped into here. FADH2 enters the electron transport chain at a little bit further down, okay, at the region of this second electron carrier, okay? And as a result, we realize less of an energy payoff from the oxidation of FADH2 than we do NADH, but the net effect is the same. The FADH2 gets oxidized to FAD, so it donates its electrons to this carrier, and then these electrons, by virtue of their physical location, begin to lose potential energy, and that potential energy is used to pump protons from the mitochondrial matrix into the inner membrane space. And so what happens is, as these electrons are passed from carrier to carrier to carrier, which are physically close to each other in the membrane, they're like almost like an assembly line. These electrons lose a little bit of energy at each step because they change their physical position, okay, like falling down steps. And that energy is trapped and used to pump protons into this inner membrane space. What eventually happens is that the buildup of these protons is relieved through this very special tunnel, this channel that exists in the inner mitochondrial membrane called mitochondrial ATP synthase. And what happens, because this concentration gradient of protons builds up in the inner membrane space, that when they reach this tunnel here, the tunnel is open and the electrons will flow down it just as a result of their concentration gradient. They'll go from where they're highly concentrated to where they're minimally concentrated. And that energy, as they flow through this channel, is trapped by this ATP synthase and used to generate massive amounts of ATP. And that's where most of the ATP in oxidative metabolism comes from. It's at the level of ATP synthase here in the electron transport chain. Now, eventually, these electrons reach what's called the terminal electron acceptor, which is the last molecule in this sequence of electron carriers to pick up the electron, and the terminal electron acceptor in the electron transport chain is oxygen. When oxygen receives these exhausted energy electrons, it'll combine with a couple of protons and it'll form water. And then the water just becomes part of the extracellular environment. As long as oxygen's around, this chain can run, okay? Because we can continuously reduce oxygen to water, okay, and maintain now a state in which the carriers in this chain remain oxidized. As long as the carriers in this chain remain oxidized, they can accept reduced compounds and oxidize them. In order for an oxidation reduction reaction to occur, one participant has to be reduced and one participant has to be oxidized. And then when they meet up, what happens is that they basically swap positions. The reduced compound gets oxidized and the oxidized compound gets reduced as it accepts electrons from the other molecule. If there's no oxygen around, what happens as these high energy electrons get donated to the chain is that the entire chain will become constipated and all the carriers will be reduced. And when a reduced molecule meets a reduced molecule, nothing happens. They just look at each other. And that'll result now in no energy payoff. And that's why oxygen is so vital for us to produce massive amounts of ATP that generate the energy needed to drive life. So the key reactions take place in the electron transport system, a series of integral and peripheral proteins in the inner mitochondrial membrane. The stoichiometry is 2 hydrogen molecules plus 1 oxygen molecule make water, okay? But it's, again, the fact that we oxidize this molecule and reduce the oxygen, that energy is used to generate now all the ATP that the cell needs in order to operate. The steps of oxidative phosphorylation involve oxidation and reduction. The loss of electrons is reduction. The reactions are always paired. When electrons pass from one molecule to another, the electron donor is oxidized and the electron acceptor is reduced. Oxidation and reduction are important because electrons carry energy as a result of their physical location. Some of this is lost as heat, but the remaining energy is used to do work, such as perform ATP production. By sending the electrons through the oxidation reduction reactions before they combine with oxygen, cells can catch and use as much of the energy as water as water is made. Okay, and so again, it's the free flow of these electrons that keeps the chain running and that keeps the proton gradient established, which is then used directly to generate ATP. Coenzymes are important in this chain. These are intermediates that accept electrons from one molecule and transfer them to the next. In the TCA cycle, NAD and FAD remove high-energy electrons from organic substrates. When a coenzyme accepts hydrogens, the coenzyme is reduced. The donor molecule loses electrons in energy as it gives up hydrogens. So it gets oxidized, okay? And again, when we're saying hydrogens, what we're really interested in here are the electrons, okay? Protons are then released and electrons enter a sequence of oxidation reduction reactions, and this is called the electron transport system. The sequence ends when the electrons transfer to the oxygen and the formation of water is accomplished. At several steps along the sequence, enough energy is released to synthesize ATP from ADP, and that synthesis happens here, okay? FAD accepts 2 hydrogens from the TCA cycle and in doing so gains 2 electrons. The oxidized form of NAD has a positive charge. This coenzyme also gains 2 electrons as 2 hydrogen atoms are removed from the donor, and this results in the formation of NADH and the release of a proton. As a result, the reduced NAD is often referred to as NADH plus a single proton. The electron transport system is a sequence of cytochrome proteins. Now, what the heck are cytochromes? Cytochromes are organic molecules that have a lot of double bonds in them. And these double bonds serve as sort of an electron conduit, kind of like wiring almost. You could think of them as hors d'oeuvre plates, and you could think of the electrons as the hors d'oeuvres. Each cytochrome has a protein and a pigment, and it's the pigment where the electrons actually sit. The protein is embedded in the mitochondrial inner membrane, and it surrounds the pigment, which contains either an iron or a copper ion. In step 1, coenzyme strips 2 hydrogens from a substrate. Okay. As we've seen, different coenzymes are used for different substrates. In glycolysis, NAD is reduced to NADH. In mitochondria, NAD and FAD are reduced in the TCA cycle. In step 2, NADH and FADH2 deliver hydrogens to the inner mitochondrial membrane. And by hydrogens, we really mean high energy electrons. Okay. These coenzymes are then carriers. The electrons carry energy, and the protons that accompany them are released before the electrons move into this electron transport system. One of 2 paths is taken in the electron transport system, depending on whether the donor is an NADH molecule or an FADH molecule. The NADH path uses flavin mononucleotide, where the FADH2 path goes directly to coenzyme Q. Okay. Both FMN and CoQ are bound to the inner mitochondrial membrane. But notice here that when we donate at the CoQ level, we miss out on some proton pumping activity, and as a result, we miss out on some chemical potential energy payoff here, and that's why FADH2 yields less energy when it's oxidized than NADH does. In step 3, coenzyme Q releases protons and passes electrons to cytochrome B. In step 4, electrons are passed along the electron transport system, losing energy in small steps as these electrons change their physical location. At the end of the electron transport system, an oxygen accepts the electrons and combines them with hydrogen ions, and then we have the formation of metabolic water. So without oxygen here at the end, the chain won't run. Okay? Concentration gradients across membranes are a form of potential energy that can be harnessed by the cell. The electron transport system doesn't produce ATP directly. Instead, it creates the conditions for ATP production by creating a concentration gradient across the inner mitochondrial membrane. The electrons that travel along the electron transport system release energy as they pass down the chain. The energy released at each step drives hydrogen ion pumps that move hydrogen ions from the mitochondrial matrix into the inner membrane space. Okay, so this is this proton pumping action that we were talking about. It's this concentration gradient that provides the energy to generate ATP. Despite the concentration gradient, hydrogen ions can't diffuse into the matrix because they're not lipid soluble. They're charged molecules. However, hydrogen ion channels in the inner membrane permit the diffusion of hydrogen ions into the matrix. That's this channel right here. Okay? These ion channels and their attached coupling factors use the kinetic energy of passing hydrogen ions to generate ATP in a process known as chemiosmosis or chemiosmotic phosphorylation. So this is why I was trying to draw the analogy to a hydroelectric plant. As these protons flow from a high concentration area to a low concentration area, that movement represents kinetic energy that is literally trapped inside this generator and is used now to phosphorylate ADP and generate ATP. So the more protons move across, the more ATP you're going to produce. For each pair of electrons removed from a substrate in the citric acid cycle by NAD, 6 protons are pumped across the inner membrane of the mitochondria. Their re-entry into the matrix provides the energy to generate enough kinetic energy to make 3 ATP. For FADH2, 4 protons are pumped across the inner membrane, and this is sufficient to generate 2 ATP as they flow back down their concentration gradient. Okay, why is oxidative phosphorylation important? Well, because it generates most of the ATP in aerobic respiration. If oxidative phosphorylation slows or stops, the cell will die. If many cells are affected, the individual dies. Oxidative phosphorylation requires oxygen and electrons. ATP generation is, as a result, limited by the availability of either oxygen or electrons. Cells obtain oxygen by diffusion from the extracellular fluid. During glycolysis, the cell gains 2 ATP for each glucose broken down anaerobically, and this makes 2 NADH molecules. These electrons are passed from NADH to FAD through an intermembrane space, and this intermediate then passes its electrons to CoQ. In the electron transport system, what this means now is that we're going to have enough electron pumping or proton pumping action to make 2 ATP. This ultimately provides an additional 4 ATP molecules because, as you remember, in glycolysis, you make 2 pyruvates for every single glucose that's split. The TCA cycle breaks down 2 pyruvates, transferring hydrogens to NAD and FAD. These coenzymes donate electrons to the transferring hydrogens. Sorry about that. The citric acid cycle breaks down 2 pyruvates and transfers hydrogens to NAD and FAD. These coenzymes donate electrons to the electron transport system. The 8 NADH yield 3 ATP and 1 water. Each of the 2 FADH yields 2 ATP and 1 water. Thus, the shuffling from the TCA cycle to the electron transport system yields 28 molecules of ATP. Each revolution of the cycle adds 1 ATP directly by way of the production of GTP. This cycling provides an additional gain of 2 molecules of ATP. Because some of the steps in glycolysis are irreversible, cells can't generate glucose by reverse glycolysis using the same enzymes. Occasionally what happens as our metabolic demands change is that we need to increase blood sugar in between meals. And the way that we do that is by breaking down our energy stores and turning them into glucose and putting that glucose into the bloodstream for delivery to cells to use as energy. This is a process called gluconeogenesis. And some of the intermediates are the same as in glycolysis, but the enzymes that carry them out and the tissues that carry out gluconeogenesis are functionally different. Glycolysis and glucose production involve different sets of enzymes, and the two processes are independently regulated. Gluconeogenesis is the synthesis of glucose from non-carbohydrate precursors, meaning what? Meaning fat. Okay? Fatty acids and amino acids can't be used for gluconeogenesis because their catabolism produces acetyl-CoA. In the liver and skeletal muscle, glucose is stored as glycogen. This is glycogenesis, which is a complex process that involves UTP. Glycogenolysis is rapid and involves one enzymatic step. So what we're looking at here is the ability to move energy stores from one type of molecule to another depending on the body's needs. Now, when we go from glycogen all the way down to acetyl-CoA in the Krebs cycle in the mitochondria, these are energy yielding steps. Okay, glycogen is broken down into glucose. The glucose can then pass into the glycolytic pathway, which generates pyruvate. The pyruvate can then enter the Krebs cycle. That generates then the high energy intermediates that go to the electron transport chain, and that produces a high energy payoff, or 36 ATP per glucose. The opposite direction is a biosynthetic reaction. And what we're doing here is that we are funneling now amino acids and lactate into the gluconeogenic cycle, and we are generating now pyruvate. That is a key intermediate in gluconeogenesis. The pyruvate can then be used to generate oxaloacetate in an energy-consuming step, which is then phosphorylated again to make phosphoenol pyruvate, or PEP. PEP is an important intermediate in glycolysis and in gluconeogenesis because it itself is a high energy molecule. It is a phosphoenol, and the phosphoenol configuration has a lot of energy stored in the bond between the phosphate group and the carbon skeleton. That molecule is then reduced to make glycerol. The glycerol can then be used to make fructose 1,6-bisphosphate, which can then be dephosphorylated to make fructose 6-phosphate, which can then be isomerized to glucose 6-phosphate, which then can be dephosphorylated, and now the glucose can enter the bloodstream and be delivered to the tissues that need it for energy. Now, how is it that fat comes into all this? Well, fat comes into all this as a result of the backbone of triglycerides called glycerol. Okay, glycerol is the molecule to which the fatty acids are joined, and the glycerol can be converted directly into phosphoglyceraldehyde, which can then be used to manufacture fructose 1,6-bisphosphate, and then from there we can generate glycogen. Okay, so that's what we mean when we say we can use fat and amino acids to generate new glucose. Gluconeogenesis, if you look at the word, gluco means sugar, neo means new, genesis means to produce. Okay, so to produce new sugar is gluconeogenesis. Now, how is it that the amino acids play into this? Well, amino acids contain nitrogen, and nitrogen can be removed in a reaction called deamination. And when that happens, there are amino acids that, when they're deaminated, can form intermediates like oxaloacetate and pyruvate, and those can be directly used to generate PEP, which from there can be used to make fructose 1,6, fructose 6, glucose 6, and then ultimately glucose. Okay, so there's a way to go back and forth between these high energy molecules in order to satisfy our energy needs. Okay, let's talk a little bit about lipid breakdown. We'll describe the pathways involved in lipid metabolism, and we'll summarize the mechanisms of lipid transport and distribution. Just like carbs, lipids have carbon, hydrogen, and oxygen, but in different proportions. Triglycerides are the most abundant lipid, so we'll talk about pathways for triglyceride metabolism primarily here. So what do we mean when we talk about lipid breakdown? During lipid breakdown, they're broken into pyruvate or channeled into the citric acid cycle. The triglyceride is first split into its components by hydrolysis, and this gives us 1 glycerol and 3 fatty acids. The glycerol enters the Krebs cycle after cytosolic conversion to pyruvate. The breakdown of fatty acids involves a different set of enzymes that generate acetyl-CoA. And so here you're looking at the breakdown of fatty acids. They're broken down into 2 carbon fragments in the mitochondrial process known as beta oxidation. The carbon chains can enter the citric acid cycle immediately as acetyl-CoA. Now, how in the heck does beta oxidation work? Well, first thing that happens if we're going to break down fat is that we cleave these fatty acids off of the glycerol backbone, and you get 3 fatty acids for every triglyceride. Now, what happens with the fatty acid is that we oxidize the beta carbon. Now, what in the heck is the beta carbon? Well, the beta carbon is the carbon next to the alpha carbon, all right? So, if you look here, all right, this is the alpha carbon, this is the beta carbon. And what we do is we oxidize this molecule so that we generate now an acetyl-CoA, all right, that can be split off from this chain, all right? 2 oxidative steps, an NAD step that results in the production of NADH, and an FAD step that results in the production of FADH2, serve to introduce a keto group at this beta carbon, which can then be used to form a new fatty acyl-CoA, and with the subsequent release now of an acetyl-CoA. Now, what happens to the acetyl-CoA? The acetyl-CoA can directly enter the citric acid cycle. I can kind of draw this a little bit for you here. Let me show you what I mean. So if we go, that's in black. Okay, so let's say this is our fatty acid. Let's say this is our fatty acid here. Make it a real short one. Okay, so there is our fatty acid. Okay, we've split this off from our glycerol backbone. Now the first thing we do is we join this to a CoA molecule. Now we're going to oxidize the beta carbon. And the way we do that is we produce now here we go. We put an OH here. That's an oxidation reaction. Okay. Then that becomes a keto group. Let's walk it up here. In another oxidation. Okay. So I was mistaken before, guys. This is the alpha carbon. This is the beta carbon here. Okay. So this is why it's called beta oxidation. Alpha, beta. Okay. And then a new CoA comes in. And forms a bond here. And that splits off now this acetyl-CoA. Okay. And now we have a fatty acid chain that's 2 carbons shorter. And where does the acetyl-CoA go? It goes right into the citric acid cycle. Okay. And from the citric acid cycle, we already know we can form citric acid, and then we can generate our carbon dioxide, our NADH, and our FADH2, and our ATP directly. And so for every 2 carbon unit that exists in this long chain, you get a turn of the TCA cycle. And so this should tell you intuitively that there's a lot of ATP payoff to be had as you oxidize these 2 carbon units. And that's a direct measurement of the calorie content of fats, the sheer number of these acetyl-CoAs that can be produced and then oxidized in the mitochondria. Each 2 carbon fragment that the cell gains gains 12 ATP molecules from the processing of the acetyl-CoA in the TCA cycle, plus 5 ATP molecules from the NADH, and the cell can therefore gain about 144 ATP molecules from the breakdown of one 18-carbon fatty acid molecule. Now think about triglycerides. How many fatty acid molecules are in a triglyceride? 3. Okay? So we're talking north of 400 ATPs for a typical triglyceride completely oxidized. This yields more energy than any other molecule that we have as a macronutrient. Lipids are needed all over the body. Free fatty acids constitute a tiny percentage of the total lipids. Because most lipids are not water soluble, special mechanisms carry them through the body as lipoproteins. Free fatty acids are lipids that can diffuse across the cell membrane in the blood. Free fatty acids are generally bound to albumin. And what you're looking at here, just the steps of beta oxidation again. Okay. Lipoproteins are lipid protein complexes that contain large insoluble glycerides and cholesterol. They're classified into 5 groups. Chylomicrons make up 95% of these. Uh, I'm sorry, 95% of a chylomicron's weight is in triglycerides. This is what's called bad cholesterol. The largest lipoproteins, chylomicrons, are produced by intestinal epithelia, and they carry absorbed lipids from the intestinal tract to the blood. Very low density lipoproteins, VLDLs, these are also called bad cholesterol, contain liver triglycerides and small amounts of phospholipids and cholesterol. The function of VLDLs is to transport these triglycerides to peripheral tissue. Okay, so what we're doing here, all right, when we're making these low density lipoproteins, is we're in lipid storage mode. Lipid storage mode. Okay, intermediate density lipoproteins are intermediate in size and composition between VLDLs and LDLs, and they contain small amounts of triglycerides and more phospholipids and cholesterol than LDLs do. Low-density lipoproteins contain cholesterol, lesser amounts of phospholipids, and few triglycerides. These lipoproteins deliver cholesterol to peripheral tissues. Because the cholesterol may wind up in arterial plaque, LDL cholesterol is often called bad cholesterol. Okay, anytime you see LDL, low density, this is the type of triglyceride intermediate, storage intermediate, that indicates that you're in fat storage mode. High-density lipoproteins have equal amounts of lipid and protein. The lipids are largely cholesterol and phospholipids. The function of HDLs is to transport excess cholesterol from peripheral tissues back to the liver for storage or to excrete them in bile. Okay, this is good cholesterol. Okay, and what this means is that we're trying to rid ourselves of the production of fat. Chylomicrons produced in the intestinal tract enter lymphatic capillaries and travel through the thoracic duct to reach the venous circulation and then the systemic arteries. Chylomicrons are too large to diffuse across a capillary wall, so that's why they have to get into the lacteals first. And so what you're looking at here is the pathway whereby we transport lipids through the body. Okay, so we'll start here at step 1. The liver cells make the VLDLs for discharge into the blood. Okay, so what we're doing here is we're breaking down fat. Okay, we're breaking down fat. In peripheral tissues, capillaries, lipoproteins, lipase remove many of the triglycerides from the VLDLs, leaving them as IDLs. What this means here is that we're breaking down fat, okay, forming these IDLs. Okay, IDLs. When IDLs reach the liver, additional triglycerides are removed and the protein content is altered, and this produces LDLs. Okay, so from IDL to LDL. The LDLs then leave the blood through the capillary pores or across the endothelium by vesicular transport. Okay, that's over here. Once in the peripheral tissues, the LDLs are absorbed by means of receptor-mediated endocytosis. The cholesterol not used diffuses out of the cell. The cholesterol then re-enters the bloodstream where it is absorbed by the HDLs and returned to the liver. Okay? And so what you see here are the HDLs absorbing the cholesterol, returning to the liver. Okay? So what does this mean when the HDLs are high? This means that you are in fat breakdown mode. You have a lot of HDLs. What we're doing is we are basically decreasing the population of these LDLs. We're using those lipids for energy, and then we're recycling the components back to the liver. Okay? In the liver, the HDLs are absorbed and the cholesterol is extracted. Some of the recovered cholesterol is used to synthesize LDLs, and some of it's used to make bile. And then finally, the HDLs, stripped of their cholesterol, are released into the blood to travel the peripheral tissues and absorb additional cholesterol. Okay, so when these guys are in high amounts, we're getting cholesterol out of the blood and we're primarily in fat breakdown mode. Okay? So when you've got high levels of these intermediates, this is good. When you've got high levels of the LDLs and the VLDLs and the chylomicrons, we're in fat storage mode, and this is generally an indication of a poor diet. Again, it can lead to arteriosclerosis.